All right, so we finished topic 8 today. We want to talk about serological tests and DNA tests, and we're going to do the first part of topic 9 today. So send an email. Just sorry for the confusion there. Just to clarify, the plan is to starting November be completely face-to-face. The other thing I found out, apparently this had changed 3 weeks ago. They put our class to 10:30 on Thursdays. So you may have noticed Tuesdays and Thursdays were different times. So apparently that was actually a mistake. They were supposed to be both at 10:30, and somebody discovered it in self-service and fixed it and didn't tell me. And I think somebody emailed me. I can't remember what day that was, Friday maybe. And I looked into it and they're like, oh yeah, sorry we didn't tell you. So here we are, Thursday, 10:30. I guess it wasn't really an issue because there was no class before us. But I think that is actually the case. There's a few things scheduled later on in the semester that would have overlapped with us. So anyway, weird things happen. I guess a little bit of extra sleep if you don't have anything before this. I don't know. So let's talk about serological stuff, okay? I showed you this slide before, and I told you we're going to come back and talk about antibodies in a lot of detail. Antibodies are part of the immune system. They are little proteins, and the little proteins your immune system makes to recognize foreign particles. Those foreign particles are called antigens. So this little yellow thing here is representing the antibody. Like I said, it's a protein. They're kind of Y-shaped. You can look them up. They're actually kind of cool. They have 2 binding sites to bind antigens, and those antigens can be quite a variety of different things. They can be things that you were maybe infected with. So maybe you had chicken pox, and now you have antibodies that recognize the varicella zoster virus. Maybe you had the vaccine for chicken pox, and so now you have antibodies that recognize antigens that were present on that vaccine, which are for the varicella zoster virus. So as I mentioned, we can make these things in animals, and typically they use mice, and rabbits are the most common, but sometimes goats, and believe it or not, llamas are used, which is a whole interesting story on one end. And they can be used to basically have these little proteins that are very specific for one thing. And so you could have an antibody that recognizes, let's say herpes simplex virus. You could have an antibody that recognizes, let's say, a protein that's found in the nucleus. And they're used for all sorts of research and diagnostic purposes. And so all these serological tests are using antibodies in some form, and usually they're commercially available. You can get these massive catalogs nowadays of antibodies and use them for a variety of different tests. So I want to show you some tests. We already talked about the immunofluorescence, which I think is the first slide here. And so this is used in microscopy. So you're getting an antibody, and the antibody has been, a fluorescent stain has been attached to it by a chemist. And so now you have a glowing marker. And so in this case here, you can see, for example, I have 3 different tests here, all with some immunofluorescence. You've got Plasmodium, causes malaria, some sort of measles virus antibody. I'm not actually sure what we're looking at here. I just thought it was a cool looking picture. I have to read that a bit more carefully. Detection of rabies virus. So a lot of these things, viruses in particular, are often found in very teeny tiny amounts, and they're very small things. And so the fluorescence can actually, we wouldn't be able to detect it otherwise, right? It's because it's glowing now, and that's giving off its own little signal. So I want to talk about some other tests using antibodies, and there's a whole variety of them, but there's some that you should know. One is agglutination. So agglutination basically means clumping. So if you take a look at this, we have, I think this is supposed to be a test tube, or that, or it's a little square slot on a glass microscope slide. And what happens is if you put the antibody in there, if it recognizes it, they clump together. So just think about these antibodies, and I'll show you a little graphic here in a moment, but they have 2 little binding pockets, right? And so they can grab onto things, and everything just turns into a big clump. And you can actually see this relatively quickly. We're talking about maybe 30 seconds kind of thing. And you don't even need a microscope, right? You put the sample on the slide, put a drop of the antibodies, give it 30 seconds or whatever the protocol says, and you can physically, with your own eyes, actually see the clumping, and you can say it's a negative test or a positive test. I'll show you some other pictures in a minute. There's different types of agglutination procedures. I'm going to show you two here. What's often done is the antibodies are attached to latex. So latex is a type of plastic. It's used in gloves and condoms and all sorts of things. So it's basically a little plastic balls is what you're looking at. And the whole idea is to give these antibodies a chance to already be attached to one another and make them more efficient. So it's just a lot faster to use the latex in this case, right? So you can see all these antibodies are attached to this latex, and there's some sort of pathogen, and so then they clump. And like I said, this isn't really, you can't really see it, but given enough clumping, you can start to see, the clumping on the microscope slide, and you don't even actually have to look at it under the microscope. You can see it physically with your own eyes. So you can see this is one pathogen being recognized, right? Sometimes the opposite is done. So notice on the first test, the antibodies themselves are attached to the latex, and we're detecting the pathogen directly. This works for anything where the pathogen is found in high enough numbers. And that's not always the case. It kind of depends on the body fluid and the pathogen. And one of the big issues with viruses, for example, in HIV, the amount of viral particles in your blood is so small that even if you were HIV positive, it actually is really hard to detect the viral particles. So instead, what we do, rather than detecting the pathogen, we're doing the exact opposite. We actually have the pathogens antigens. So in the case of an HIV test, the antigens would be a glycoprotein found on the surface of the virus, and we're actually going to detect antibodies in the bloodstream. Because if you have HIV or a variety of other viral infections, your immune system is going to make antibodies. And the antibodies are actually going to be far more numerous than the actual virus. And so what we're doing is we're actually looking for antibodies in the blood serum. Right? And we're going to use that detecting the antigens. So it's kind of the opposite idea. And same idea, you're still getting some clumping. So these are used for a variety of different types of tests. Like I said, it kind of depends on the pathogen and the body fluid and all those kind of things. And they're pretty efficient and easy to use. I think I have some examples here. I think I told you last day I was going to talk about the streptococcus a little bit, and how there's the group A and the group B, and it goes all the way to, I think, group L or something like that. And this was actually based on these agglutination type tests. So if you take a look here, this is the group A streptococcus, right? Streptococcus pyogenes. This is one of the ones we need to know for this class. And it's a little hard to tell on the slide. I think I've got a bit of a zoom in. There's the detail. And you can see that the positive, if you look carefully, maybe you can see the clumping a little bit better. Sometimes it shows a little bit better on my monitor than the actual big monitors here, but that's why I zoomed in, and you can see it's a lot smoother in the negative test. So in this case, what they're doing is they have an antibody, and the antibody binds the group A streptococcus. And when it does, they start clumping together, and I'm not even sure in this case whether they use the latex version or not. Here's all I'm not even sure if this is all, but this is a bunch of the group streps. They're called the Lancefield grouping. And you can see it, I thought it went to L. Obviously, it goes further than L. I don't know who Lancefield was and why he didn't actually look at Streptococcus pneumonia, but Streptococcus pneumonia is not on this group. So I added at the bottom as a non-Lancefield Streptococcus. Sometimes it's referred to that because it was something he never characterized, which is, like I said, is really odd because it's a really common human pathogen. So I don't know really his background. But the two in the red, right, those are the ones you need to know. But you may encounter in your travels some of the other group strep. One of the ones that comes up a lot is the group B strep, and that's pretty common in prenatal situations. Women are tested for it to see if they have it because it is a risk to the newborn. Okay, some more pictures here. You can see there's kind of a test, right? So in this case, you can see it says they took some spinal fluid and they're using the agglutination testing to diagnose which one it actually is. So if you take a look, you've got A, B, C, D, and E, and B. It's pretty obvious the clumping is in B, and in this case, it's a group B streptococcus, I'm guessing. I'll just have to read the, just checking by reading the caption down there. So one more thing I want to show you is, oh, sorry, getting ahead of myself. It's not just used for streptococcus. There are other tests used for bacteria. This was a staphylococcus test. Just wanted to show it to show you that it's not just streptococcus. One thing you may have done, I did this. Well, I didn't do it. My partner did it in grade 11 biology. I don't think you're actually allowed to do this anymore, but we did blood testing. I think nowadays, they don't want to test people's blood in high school due to risk of diseases and just in case you find out you're not related to your parents or something like that. But there's a lot of good reasons for not doing it. But my partner did it, pricked his finger and put it on a glass slide and did blood typing. And this is how blood typing is done. You just need 1 drop of blood and you're adding antibodies. And so you may know that there's the A, B, O blood groups. And so there's that actually turns into 4 different types of blood groups. You can have A type, you can have B type, you can have AB, or you can have O. And so I want to show you how this works. It's the same idea. You've got some sort of thing that you're looking for. In this case, it's not a pathogen, but it's somebody's red blood cells. And we have antibodies, and you're going to end up with clumping. But in this case, we have 4 scenarios, and I'll show you how this works. If you have, and what they do is they do 2 tests actually, so 2 drops of blood, 1 drop and 2nd drop. And on the 1st drop of blood, they add antibodies that recognize the A antigen. And on the other drop, you have antibodies that recognize the B antigen. So what are those antigens? Those antigens are actually carbohydrates that are found on the surface of red blood cells. And there's just a variety of different types in the human population. And depending on what your parents gave you in terms of the alleles, you can have A only, you can have A and B, you can have B only, or you can have neither, which we call O. And so you can see these scenarios. If you're AB type, you're going to see clumping on both drops of blood. If you're A type, you're going to see clumping only on the with the A antibodies. And if you're B type, you're going to see clumping only with the B antibodies. If you're O type, you're going to have no clumping at all. So very, exact same test, just not testing for pathogen. It's really cool to see this. I don't know if anyone has seen this, but it's very, very fun to watch it be done and find out what your blood is. I don't actually know what my blood type is today because I never did it, got my partner to do it. I'm sure somebody knows. Okay, there's one other type of test I want to talk about that is gaining a lot of popularity. And you may have heard about these rapid tests that are done with COVID and whatnot. And they're all based on this principle called an ELISA. So an ELISA, that's an acronym. It stands for Enzyme-Linked Immunosorbent Assay. So lots of words there, but you can see immuno is in there. And immuno means usually has something to do with antibodies or the immune system. Assay means test. And enzyme-linked is the thing that is going to make this detectable. So I'll show you how this works here in a second. But usually these ELISA tests are done in these, we call these a multi-well plate. And so there's different versions of these plates, but the standard one has 96 little wells. So it's kind of like 96 little test tubes or 96 little places in a tray. So you can do 96 tests. And I'll show you how this works and what the enzyme is all about. So here's the little well in the plate. And usually the first step is adding whatever the sample is. So it could be saliva, blood, whatever the test is going for. And in HIV, this is usually blood when they're doing HIV testing. And this is actually one of the main tests used for HIV. If people do a rapid test and they get a positive, the rapid tests are not 100% accurate. And so the ELISA is sort of the backup test, even though they're kind of based on the same thing. So you can see there's the first step. You throw in the sample. The plastic is actually coated so it's very sticky. And you throw in the sample. It actually goes through a washing step. And then we throw in the antibody. Okay, so the antibody is recognizing the antigen. Now in HIV, it's the opposite. We are actually looking for antibodies, but it's the same principle. And it's really the second step that is the most important. I'll show you in a second. So basically, you have an antibody recognizing an antigen. That's great, except for we can't see anything, right? These things are microscopic, and so we want to be able to see them. So what we actually have is a secondary antibody. I know this is kind of like, why did you do the second antibody? Why not just do something with the first one? Well, the classical methods were radioactive, and we don't like working with radioactive materials. And the other thing is you could get a chemist to modify every single antibody out there. It would be very expensive. You know, like I said, we have these massive catalogs with thousands of antibodies for thousands of tests. It's just kind of crazy to do. So instead, what we do is we have a secondary antibody that actually recognizes the first one. And the key thing is there's an enzyme on there that's attached, again, by a chemist. And the enzyme is going to help us to see what's on there. So the enzyme, you know what an enzyme is, it catalyzes a chemical reaction. And this chemical reaction produces a color. And that's actually what we're looking for, is a color. Okay? And so I'll show you what this kind of looks like. Right? Is you have this plate, and you have all your samples. You're going to have blanks. You're going to have controls, negative controls, positive controls, and then all your samples. So where you see the color is where you have a positive test. So, you know, all these yellows are people that are positive for whatever we're testing for here. And it can actually be read by an instrument and usually verified by a human as well. But it's actually a very easy to do test in terms of technician can do this relatively quickly and kind of have you know, responses, you know, answers within about 30 minutes. And it's highly reliable test. So you do not need to know all of these steps, by the way. Okay, I'm not going to ask you about all these steps here on a test. For the test, what's going on here? Go back to the computer. For the test, what I want you to know is basically kind of a definition, right, of what an ELISA is. Okay, so you know, I could see a multiple choice question or something like that. So you need to know it's an immune kind of a test, right? It's using antibodies, and it results in you know, a colored response that's a yes or a no. Okay, that's kind of what I want you to know for the exam. So what's going on here? So these are done by technicians. Okay, it requires a little bit of know-how, you know, pipetting expertise and whatnot. But what's gained a lot of popularity, partly because the accuracy of these things has improved dramatically, are these rapid tests. And I'll show you some pictures of some rapid tests. I actually have a rapid test right here, but it kind of looks like the one on the screen. This one's for Legionella, and the flyer is one for influenza. And like I said, there's a ton of these things now that you can get, and they've gained a lot of popularity. The problem is they never used to be that accurate, right? And by not that accurate, they're about maybe 80% accurate, maybe 90%. That doesn't seem too bad, right? Like a lot of people are very happy with an 80 or 90 on a midterm. But if you think about it, if something is 90% accurate, that means 1 out of every 10 people is going to get the wrong result. And that's actually pretty bad. So usually they used to be used as kind of a preliminary quick test because these things take anywhere from 5 to 20 minutes, right? And so that's a huge advantage. Take a look. I have this one for HIV, and some people are probably looking at this and realizing it's very similar to a pregnancy test, and actually it's the very same technology. And so the way it works is usually there's a little place. It's I don't know what you want to call it, a card or whatever. It's a plastic thing, and there's a little place to put the bodily fluid. So in the case of a pregnancy test, and I'll show you a little video that actually shows the pregnancy test and how it works. But basically, the bodily fluid is put there. So it could be urine in case of a pregnancy test, blood in the case of HIV or saliva or something like that. And then the fluid will actually migrate through the test, and there's antibodies and colorized agents in there. And that's how it's actually being recognized. So if you take a look at the HIV test here, for example, if you get 2 bars, so the C stands for control, So the control bar is just, it should always go off if the test is working properly. So in the case of like the third and fourth test, you can see the control didn't go off. So that means that this particular kit is expired or something like that, right? So the control should always go off. If you get 2 bars, it's a positive. If you get 1 bar, it's a negative. So I'll show you how this works in a moment. But the accuracy of these things has gone up. The HIV tests originally were 80%. I think now they're better than 95%, and still not that great. If they're 95%, it means 1 in 20 people could be HIV positive and they don't know it. But huge advantage of these because you get immediate results, and in many cases with some of these tests, people, they may not come back, right? Think about an HIV test, right? This could be at a clinic for, let's say, drug users, and they want to know, and you could take the test, and they may not come back in 2 days to get the results. In this case here, they can get the test within 10 minutes and start counseling or thinking about what their next steps are going to be right away. Usually the backup test is done as well, just to make sure. But like I said, huge advantage of these rapid results. So they've been talking about this a lot because now there's a bunch of these rapid COVID-19 tests out, and it's the same technology, right? And I think, I'm not sure if he's using saliva or not. Anyone know? Is it saliva? They put it in your nose? Oh. Every time I think about that, I just... No? Yeah, okay. Well, the PCR tests are in your nose, but the rapid tests, are you sure, you know, that's in the nose as well? Yeah, so they've got to get enough liquid out of there. Oh, okay. Yeah, I'll show you the video how this works. It's really cool. I found there's actually a few of these on the internet now, partly because of COVID-19. But the best video I found actually is the one on the I have one on the pregnancy test, and I'll show you that one in a moment. But this is kind of showing a little diagram of what's going on, and there's little antibodies in there, and I'll show you the video and show you how it works. There we go.
A pregnancy test works by measuring a specific hormone in the body, human chorionic gonadotropin, or HCG. About 6 days after fertilization, the human embryo has multiplied from 1 to 2 to 4 and eventually into a multicellular organism called a blastocyst. The blastocyst then implants or attaches to the lining of the uterine wall or endometrium. After implantation, the body begins to produce the hormone HCG. HCG levels rise fast and can be measured either through a blood test or a urine test. Home pregnancy tests use urine samples to test HCG levels. The pregnancy test contains antibodies that bind to HCG, which is connected to an indicator molecule that is usually a pigment, the pigment antibody complex. When the HCG enters the region, it selectively binds with the antibody, thus activating the indicator molecule. The pigment is revealed in a line across the region indicating a positive result.
Okay, so pretty cool technology. Like I said, not completely 100% accurate, which is usually why a follow-up test is done. But the advantages are that you have answers right away. Like I said, depending on the test, could be 5, could be 15, 20 minutes. And like I said, the classical one is the pregnancy test, you know, and you don't have to take a blood sample, you don't have to be trained, so lots and lots of advantages. Okay, so the last part of testing I want to talk about are the genetic characterization. These go by, you know, these things used to be so simple, but they've been going by so many names nowadays. So trying to, you know, talk about what they are is getting extra complicated. But 2 of the main names that I'm seeing out there are PCR. That's what they used to be all called PCR, but now there's a few kind of tests that are sort of not really traditional PCR. So now they're just being called nucleic acid tests or nucleic acid amplification tests, of which PCR is the most commonly used method. So I want to talk a little bit about PCR and how that works. So PCR stands for polymerase chain reaction. So polymerase is actually DNA polymerase. And I'll show you how this works. It's a pretty cool idea. And it's used in hundreds of applications and diagnostic tests and scientific tests and whatnot. I don't even know how many PCR reactions I've done in my past research, probably hundreds. I have no idea. It's kind of hard to count. It's just such a useful technique. So you can see in this case, there's a little graphic talking about what's done for SARS-CoV-2, the SARS coronavirus. And in this case, so you get the test. They take the thing that goes up your nose, the nasal pharyngeal swab, and they're trying to get some fluids, and they believe that's where the virus is found. Interestingly enough, I've seen some research to show that may not be necessary to go that deep in, but they're still doing it, probably because you have something that works and is reliable. You're not going to change your methods. So in this case, what you're looking at is an RNA virus, right? So you're detecting RNA, okay? But we're going to talk a little bit about DNA, but the principles are the same. So you get the RNA extracted, it goes into a test tube and gets amplified. So I'll show you how this works. It's pretty cool. So PCR works by basically doing what a cell does, but you're doing it in a test tube. So a cell, to copy DNA, has a DNA polymerase and a whole bunch of other enzymes, and it's got nucleotides, and so it's doing this whole process, right? So this idea behind PCR is like, well, why don't we just take a polymerase and nucleotides, put them in a test tube, and let them do their thing? Now you can't. It's not that simple. DNA, you probably know, is double-stranded, so the two strands are held together by hydrogen bonds. And so what you do is you melt it. Literally, you melt it with heat. So you can see that very first step is heated to 94 degrees Celsius. So two strands of DNA melt apart. You throw the enzyme in there, and the enzyme and the nucleotides, they make another strand of DNA. So you start off with one strand, and then you end up with two strands, right? So that's great. That's not a lot of DNA. But you do that again and again and again, right? So go on your calculator and go 2 times 2 times 2 times 2 and do that 30 times, and suddenly you end up with millions of copies of DNA. So if you think about it, you could have a very tiny, tiny amount of viral particles, and now we can detect it. And very, very powerful method, very, very sensitive. This is why you see, like on crime shows, right? They find that one strand of hair, and that's enough to do a genetic characterization of it. And same thing with diagnostic purposes in medical uses. You don't need a lot to be able to detect it. Sometimes actually this is so sensitive that you do end up with false positives, and that happens once in a while. But the techniques have really improved dramatically. You can see I have some other methods mentioned here. Forensics is an obvious use. People usually see that on TV, and the same technology for paternity testing. I've done a ton of PCR for genetics and biochemical research. It's very, very useful for a variety of things. There's a lot of paleobiology now, extracting mammoth DNA and whatnot, some really super cool things. Of course, we're mostly just talking about detection of pathogens. So why would we use DNA tests? We use them when something is found in small numbers. So that includes certain viral infections, things that grow slowly. So tuberculosis, there are PCR tests for tuberculosis, and some things we can't actually culture in a lab, like chlamydia. So chlamydia, we don't know how to culture that organism. No one's figured it out yet. So it's a urine test, and they test the urine for the presence of the bacterial DNA. And so it's has its uses. It's a little expensive. Usually you can't do it in one day. I mean, you could, but the test results are usually going to take 24 to 48 hours. And when you're backlogged, I think a year ago, I ended up getting a COVID test, and it took like 8 days, right? It was kind of frustrating because, I mean, if you think about the quarantine period, I basically was almost done by the time I got the test results back. So it is slow, a little more laborious, but it's very, very sensitive and works really well. I was just going to show you this here, how some of these tests work. There's a variety of different methods. There's a lot of fluorescent methods now that are used for pathogen diagnostics, but classically we use these things called a gel. If you take a look at this, what you're looking at, this is actually mother, child, father, right? And so in case of a paternity test, you're looking to see, does the band match the parent? Now, in the case of a child, right, the child is going to have half of his or her DNA from mom and half of the DNA from dad, right? And so you should see each child, 50% of the bands should line up with mom, and 50% of the bands should line up with dad. I'll show you. There's another gel here, and it says here, which of these children belong to this father? Basically, is what it says. So the whole idea is you're looking to see, okay, does the profile match 50% with the father, right? So in the case here, child 4 is a yes, child 2 is a yes, child 3, child 3 down here, child 1, okay, all good. They all belong to this dad, so that's good. The real tests are a bit more complicated. You're looking at multiple bands, and if you've ever watched movies and they show them, they always have them nicely colored, but they're never colored, not in real life. Okay, so by the way, these molecular tests and these serological tests, like I said, are what are being used for COVID-19. I found this pretty decent video here. I'm not going to play it. It's 9 minutes long, but I included it in the PowerPoints for anybody who's looking for a little bit more information on specifically the COVID-19 tests. Okay, so finish off this topic with a case study. So you can see it says we have a 15 or 17-year-old female, and she's nauseated and weak, and eventually it gets worse. Fever, confusion, stiff neck, and a rash. So some of these things already, anyone who's seen this before probably has a good idea of what it is. But we'll continue to go through this because you can see there she gets brought to the ER. She's having problems with consciousness. They take some blood samples and spinal fluid, and you know, same night in the ER, an 18-year-old shows up, kind of with some of the same symptoms, right? And so they sample his spinal fluid as well. And you know, talking to the patients, they find out they were at a New Year's Eve party, okay? And they do the gram stain of the spinal fluid, and you get gram-negative diplococci. Okay, so already, like I said, if you know what's being talked about here, it is super obvious. But this is what we want to talk about. But the answer is that we're actually looking at Neisseria meningitidis. So you may remember a few topics ago, we had gram-negative diplococci. What was it? You remember gram-negative diplococci? It was gonorrhea, yeah. So Neisseria gonorrhea is a related organism, gives the same gram stain, but is associated with, like it's a sexually transmitted infection. This is associated with spinal fluid, and these symptoms, you know, the rash and the stiff neck and things like that, those are very characteristic of Neisseria meningitidis, which causes a type of meningitis, right? So meningitis is inflammation of the membranes around the brain, okay? So some questions about this. It says here, how is Neisseria meningitidis spread? Anyone know how it's spread? Can be saliva, yeah. So there's kind of two things that go on here, right? So, okay, New Year's Eve party, right? What sometimes goes on at New Year's Eve parties? Anyone know? Kissing, yeah. So apparently that's a thing. I don't know if I missed out on those ones, but you definitely see it in movies, you know? It's like, oh, 12 o'clock and everyone's suddenly kissing, right? I've seen that in movies. Saliva, yeah. So it can be part of your normal flora. So sometimes you can self-infect yourself, right? It can be just found on your saliva, it can be found on your skin, and it's just a matter of it getting into your spinal area and, you know, through cracks or whatever, okay? But saliva spread is pretty common. and New Year's Eve party. So this is the kind of thing that, you know, I don't know whether those two were kissing or they were kissing somebody else, but that's actually a follow-up question here, I guess. The fourth question there, you know, what kind of follow-up should be done? You know, maybe we need to ask these teenagers, okay, do you mind telling us who you kissed? We might want to follow up with those people, right? So spread by saliva, or like I said, sometimes you have it as a part of your normal flora. So meningitis, I think I answered that already. It's inflammation of the membranes that surround the brain and spinal tissue, and can cause fever and confusion and you know, a bunch of things like that. It can be quite serious in some cases. So it needs to be treated, and this can be treated by antibiotics in this case. What other organisms? I guess we mentioned that Neisseria gonorrhea is also gram-negative Diplococcus. There are others, but those are kind of the main two gram-negative Diplococci that affect people, human health. Like I said, there's a few others, but they're just not that common. So these are the two that are really worth knowing. Last question, what protection strategies are there? Anyone know how we can protect ourselves against Neisseria meningitis? Most people don't know their status. So there's actually another protective method, which I am too old to have had. Again, it's a vaccine. This is part of a normal childhood vaccine that came out, not sure what year. I have a nephew that had meningitis, and I think he was just like a year or two old to have been included in that as getting as a baby, but his younger sister had the shot, right? I remember my sister talking about it, saying, oh, too bad Jacob didn't have this and all that. But so there is a vaccine for Neisseria meningitis that they give you as a child. So that's a theme for a lot of infectious diseases, of course. Okay, that is it for topic, whatever topic that was, 8, I think. All right, time check. All right, I guess I'm officially about half an hour behind in medical micro this semester. Not going to try to speed things up, but maybe make some ground near the end of the course. But I do want to make sure I cover the appropriate material properly. As thinking way ahead, I may have some extra review session or two at the very end of the semester if I feel like there's a bit of a need for it. And that will be totally optional, but I'm not going to rush anything on this. Topic 9 and 10 are a bit longer topics. Both of them are usually about 2 lectures each. And then we'll have some review for the midterm, and then we'll have the midterm. So that's November 2nd. So I think we're 2 weeks away from November 2nd, a Tuesday or Thursday? Tuesday, I think? Yeah, okay. So we're exactly 2 weeks away from that. So just a reminder. So topic 9, we're going to talk about sterilization and disinfection. And the whole idea here, you can see sort of the subtitle is controlling microbes in the environment. Okay, so this is something important to think about because, of course, we're trying to do them all the time, and we, you know, of course, we're talking about this all the time now with these wipes we have, you know, these Lysol wipes and hand sanitizers and things like that, and so, you know, what's effective and what can we use, and so this is what we want to talk about in today's topic here. So first, some definitions, okay? Drives me nuts how often these things are misused, these definitions, in terms of people not understanding what sterilization is versus disinfection, for example, right? So sterilization means you're killing everything, basically killing everything. Prions, maybe not. They're super hard to kill, and we're never testing for them. And they are actually resistant to a lot of methods, believe it or not. But basically, you're killing everything. Okay, and disinfection means you're killing as much as possible. Okay, so I think I have some definitions in your notes on that. So sterilization usually means everything is absolutely killed, although the prions might be the exception. Disinfection usually means you are cleaning a surface with some chemical. That's usually the scenario of what is going on. So you can imagine if I were to wipe down this bench here with some sort of strong chemical, it would probably kill most or everything, but maybe my wiping technique isn't perfect. I'm missing a spot, or 5 minutes later, after the chemical has evaporated and dried up, you still have stuff coming in from the air, dust particles and whatnot. So disinfection is kind of like hopefully you got most of them, but the methods are not perfect. And like I said, usually we're talking about chemicals. So disinfectants, we're not going to get to today. Today we're going to talk about some sterilization and some physical methods of dealing with microorganisms. Couple other definitions for you. Sanitation kind of falls into that same category of some stuff we're trying to do to kill microbes. Sanitation usually we're talking about kind of public health stuff, right? So think about like water treatment, right? Water treatment, they're doing things at the water treatment plant. They're adding chlorine, they're adding potassium permanganate, they're filtering particles out, and the whole idea is to reduce the number of microbes so that there's a very, very tiny amount. They're not going to get rid of all of them. And of course, that water is going to go into the pipes, and by the time it gets to your tap, who knows what the plumbing is like in your house. But if they've done their job well, the number of microbes are very, very tiny amount and not going to cause any health issues. You can think about things that go on in all sorts of industries. Restaurant industries have dishwashers that wash at extremely high temperatures, et cetera, et cetera. So that's sanitation. Antisepsis. So what is antisepsis? Usually when we say antisepsis, we're talking about basically disinfecting someone's skin, right? So in the case of, for example, giving someone a shot, get the little alcohol rub on the shoulder, that's a disinfectant. And then you give them the shot, hopefully minimizing the number of microbes that might actually go through that puncture. And one more, degerming. Degerming is kind of just getting rid of as much as you can. And so usually we're talking about mopping something up or washing our hands, where the whole idea is we're just kind of cleaning things up, not necessarily killing them. So we'll talk about degerming in a bit here. I'm not sure if we're going to get to that today. Okay, so before we get into the methods, I want to talk about how we kill these things. And we kill these things by basically affecting different parts of the cell. So one big area is the membrane, and the membrane, of course, surrounds the entire cell. And so this is a great target, and the membrane is made of phospholipids. So anything that affects phospholipids or phospholipid bilayers is going to kill that cell. So in the case of, for example, hand sanitizer usually uses some sort of alcohol, and the same alcohol that people might consume in their beverages. And so small amounts of alcohol, most people can tolerate, 1 or 2 drinks, not too bad, because we actually have enzymes that break it down. And when we start consuming too much alcohol, it does start getting into our membranes, and particularly in humans, it can get into membranes and neurons and kind of affect the transmission of neural pulses. And so you probably know if you've had too much alcohol before, your thinking gets slower, your reflexes are slower. But at high amounts, it actually can kill cells and can be toxic, which is alcohol poisoning. So it's actually a really effective, a reasonably effective disinfectant. It gets into membranes and basically dissolves them to a certain degree. And that's what's showing on here. So we'll come back to alcohol, like I said, next day. So membranes are one thing we can target. We can also target proteins. So proteins, of course, are, many of them are enzymes involved in essential processes in microorganisms. And we can kill proteins by chemicals. We can denature them using heat. We can denature them using different pHs. Again, a variety of methods to destroy proteins. Once they don't have their enzymes, they can't live, and that's what we're trying to do, trying to kill these things. Last thing we can do is we can target nucleic acids. And kind of the main thing we're using to target nucleic acids, in particular DNA, is actually using radiation. So UV sterilization and whatnot. And so I'm thinking this is where we're going to probably get to today. So something else too is how effective are these methods? And it actually depends on a whole bunch of things. One is it depends on the actual microorganism. So we've talked about some of these things already. Things like endospores are super tough, right? Endospores can resist a lot of heat. They can resist a lot of chemicals. They can resist drying up. They're very, very tough. They're not as tough as prions, but they're kind of top of the list in terms of things that are really hard to get rid of. And so endospores are kind of the gold standard. If this method can kill endospores, it's a good, better than good, it's a great method. Like I said, prions are not usually a concern, and prions are very hard to detect as well, and not very common. So endospores, if we kill endospores, it's an awesome method, basically is what we're saying. You can see everything else in there. Remember the mycobacteria, that's tuberculosis. It has a waxy layer. It's a little bit more protective. We also talked about cysts that are formed by protozoans, and they're kind of like endospores. They have an extra kind of wrap layer on there, and they're resistant to drying and things like that and whatnot. And you can see everything else is sort of just getting, you know, kind of weaker and weaker down there. You can see gram-negatives are apparently better than gram-positives. Probably, again, an extra layer, an extra membrane. And envelope viruses are the most sensitive. So remember, I was just saying a minute ago, envelopes are, which is, of course, the membrane, can be quite sensitive to chemicals. So this means that envelope viruses are more susceptible to hand sanitizers. So hand sanitizers, for example, do they work on coronaviruses? Yes, coronaviruses are actually envelope viruses. Do they work on norovirus? No, they don't. Norovirus does not have an envelope and is not going to get rid of it by using hand sanitizer, for example. So it depends on the nature of the organism, the biology of it, and hopefully that makes sense. What else? The numbers. If you have, you know, let's say 10 million bacteria in a contaminated water sample, that's bad, right? If you have one bacteria in there, it's probably not going to make you sick. So we can get rid of most of them. That's a big part of the job, and we'll talk about degerming. Sometimes there's other environmental things. You can imagine in a hospital, you know, somebody vomits, let's say in the waiting room. There's going to be mucus, other debris in there that may prevent the disinfection or the cleaning up of it, which is why you got to get rid of all of it, right? And exposure time. So this is more relevant to chemicals, but it's true for a lot of these things. The longer you hit the organism with something, the longer you hit it with a chemical, for example, the more likely it is going to kill it, and so on. Same thing with heat and radiation. So I think I mentioned this organism once before, and we're going to start talking about it here and there a few times. This is Clostridium difficile. So difficile means difficult, and this is Clostridium is the organism. And so this, the Clostridium organisms, by the way, are notorious for a couple of things. One is they're notorious for making endospores. All right, so when you hear endospores, you should think, okay, this is going to be one tough bug, right? The other thing that they're pretty famous for is being anaerobic, the Clostridium organisms. So here's C. difficile. On the left, you can see the endospores forming. There's a bunch of acronyms for it. Most people I know call it C. diff, right? But sometimes I've seen acronyms on sheets where it's CDF, right? So Clostridium difficile. So what's the big deal with this guy? It's difficult to kill. And so this is something that we're concerned about, and we want to, you know, when we're doing treatment or when we're doing disinfecting, things with endospores. And this one is spread in the feces, and so it can be a hospital-acquired infection, and people can get it from staying in a hospital. And these endospores, like I said, they can be very resilient, and of course, if you're transmitting to other people, this is a serious deal. So this is one of the reasons why we're trying to kill things, and we're trying to do it in a really effective way, because we're trying to prevent infections like this. So we're going to come back to C. diff a bunch of times over the next few lectures. It's one of those things that 20 years ago nobody really talked about, but now it's kind of like the #2 superbug, right? So it's a big deal. It really is. And if you ever in your practicums experience C. diff, you will know. Apparently, the stool has a really bad rotten egg smell to it. So apparently, it's pretty obvious when somebody has one of these infections, and not pleasant at all. Okay, so my goal today is to basically cover physical methods for killing microorganisms. And so by physical methods, I mean non-chemical, right? So things like heat and radiation and filtration are kind of the main things we want to talk about. And the heat is the big one that we're going to get into. So I kind of have a little, try to color code this. You know, I like colors. I like to color code things, like things to be organized. So what I want you to do is when we go through these is think a little bit about, first of all, you know, what are the most ideal methods? Okay, because some of these methods I'll be talking about, I'm going to say this is maybe not the most ideal in a healthcare setting. And the second is where they're actually used for. Okay, so and I'll kind of get into some of those examples as well, right? So let's talk about heat, right? So what does heat do? Heat can melt things. So the scientific term we give for that is denatured. So proteins, for example, are these nice folded macromolecules, and when they get denatured, they kind of just unravel, right? So if you've ever cooked an egg, that's actually what you're doing, right? So in the egg, the liquid in the egg, it's loaded with proteins. And those proteins are soluble, and they're forming kind of a, I don't know what you want to call it, a goo or a gel or whatever. But when you heat them, the proteins get unraveled, they get all tangled up, and they become sort of a stiffer gel, right? And so, you know, a cooked egg, of course, is a lot less liquidy than a non-cooked egg. And that's actually what's happening when we denature. So you're killing the proteins in this egg, is what you're doing. And this kills microorganisms. And this is, like I said, a really useful method. So let's talk about how we can use heat. We can use heat for pasteurization. So what is pasteurization? It was named after Louis Pasteur. Going way back, way back to lecture 1, we talked about Louis Pasteur, and I mentioned he'd done a whole bunch of things. One of the things that he started looking at as a microbiologist is what caused food spoilage, and learning that food spoilage was caused by microorganisms. And this was a huge leap, and I think I mentioned before, understanding that food spoilage could be caused by microorganisms, and so that maybe microorganisms could cause problems with humans, so the germ theory. So he experimented with some methods, and of course, you know, the whole concern with some foods, if you think about juices and beer and milk, you could heat them up and kill the microorganisms, but sometimes the taste isn't as good, right? So they invented a method called pasteurization, which basically means you're hitting it for a short amount of time with heat, and hopefully it's not going to affect the taste too much. Now, some people might argue and all that. I don't know. I don't drink unpasteurized milk, so I can't answer that question. But you know, maybe ask a dairy farmer if you know anybody whether there's a difference or not. But that's the whole idea, and it's actually required for a lot of foods in Canada that these things get pasteurized. So we're looking at liquids. Mostly we're talking about beverage industry. Some medical liquids are pasteurized. And you can see there's a bunch of different protocols in terms of, you know, in terms of how much heat is subjected to the liquid. So, you know, usually what happens in pasteurization is it's going through a tube, right, in a factory or whatever, and for a short amount of time, the heat hits a certain length of that tube, which is equivalent to a certain temperature for a certain amount of time. So it may not kill everything. There might be some thermophilic organisms or endospores that might survive the process. And this is why the food industry has best before dates for things like milk. And so, you know, it's kind of their best guess as to when those thermophilic organisms or those endospores might start growing. And so I used to have in high school a friend, and her parents owned like a corner store. It was kind of interesting because they only bought the corner store when she was like 16 or something, right? And she was telling us that like once the food hits the best before date, they can't sell it anymore. And she was telling us she knew in her head, she had knowledge of all the foods and which ones were good after the best before date and which ones weren't. So she was saying like chocolate milk would go bad on the day of, whereas white milk, she said, was good for about 4 or 5 days, as long as it wasn't open, kind of thing, and whatnot. But like I said, it's a best guess. And you know, some people are really sensitive to best before dates. You know, they're going through the shelves looking for the more recent ones, and so on. Okay, so let's talk about what's useful for medical stuff, right? Pasteurization, like I said, some things are pasteurized, some fluids and whatnot. but not as common. Boiling is an old classical method, again used for food preparation. If you've ever made jelly or jam or canned food, then you probably try to sterilize the jars by boiling them, and the food itself is cooked. It may not kill everything. So boiling is not a gold standard. Boiling is like if you're out in the field and it's an emergency, you boil things, right? But for medically things, we're going to use something better than boiling. And because of this reason here at the bottom, you can see it may not destroy endospores. And that is a big deal. So, oh, I don't know why I have this slide twice. But there's C. diff again, reminding us you don't need to kill those endospores. So how do we kill endospores? We use something called an autoclave. So what is an autoclave? An autoclave is an instrument, and it provides heat and moisture and pressure. So you can kill endospores if you boil something long enough, right? But who wants to boil everything, every instrument and whatnot in a hospital for like half an hour or whatever? You know, it takes time and it may not be successful. But whereas an autoclave uses pressurized steam, so the pressure is what makes the steam actually hotter on a physical level. And so usually the pressurized steam is at, I think it's 121 degrees Celsius, but with the pressure, its effective heat is actually a lot hotter than that, whereas boiling water is only about 100 degrees. And so this is how we kill endospores, and this is actually the gold standard, okay? So if I ask you how to sterilize something on a midterm, autoclaving might be the answer, and it actually is the right answer for many things. So what are those many things? Solid objects and solutions, and anything that's okay with getting wet, right? So I think I have a picture here. You can see here's an autoclave in a hospital setting, and it's kind of hard to tell what all those things are, but you can see we're looking at bed sheets, so a lot of instruments like scissors, scalpels, those kind of things can be autoclaved. You know, a lot of those tools that you may have had put in your mouth at the dentist, right? The dentist probably has an autoclave. Sometimes they do 2 methods of sterilization at the dental office. But autoclave is what they do. They stick them all in there. We can also talk about liquid things, right? So all those agar plates that I've made, usually they're produced by putting the media through an autoclave and so on. In the lab, you'll be using test tubes and anything that's okay with getting wet, right? You can dry it out later. And like I said, you can sort of make out a variety of objects there. So that's a relatively large autoclave. Here's one of the college. We actually have 3 autoclaves here. This is a benchtop one. We just don't often have massive amounts of stuff to sterilize. And so this is a benchtop one. You can see you got to crank the door shut because that has pressurized steam in there, right? We don't want this thing to start leaking and have hot steam spitting out the side or anything like that. Tons of these things. Looking for pictures of autoclave on the internet, and apparently they use it at NASA. I guess that's part of the process for making satellites. They put them in an autoclave and it seals the gaskets somehow. I don't know. I just thought that was kind of interesting. Massive autoclave there. Autoclaving is the gold standard, but you can't autoclave everything, right? I think one year on the midterm, I asked about somebody had a contaminated bed mattress, right? And people were telling me to autoclave it. Well, I mean, most hospitals have something like this or smaller. You're not going to stick the entire bed in there. Or how do you, clean a room? You're not going to dismantle all the cabinets and stuff, right? So autoclaving can't be used for everything. But anything that can fit in there, that can experience heat and can be okay being wet, right? Because we're talking about steam, is okay to autoclave. And that's usually what is done for a large majority of things. By the way, the autoclave we have here at Kano, and this is standard everywhere, you have to test them to make sure that they work. And the way that we test them is actually we use endospores. So you can buy these little vials, and there's different varieties. Actually, the ones we have turn pink or purple, and they start off as not quite yellow, but another colors aren't quite right here. But basically, you have a little vial, and it has endospores in it. You stick it in the you stick it in the autoclave, and then you incubate it for a bit. And if it changes color, it means the autoclave didn't work. If it stays yellow, then it means the autoclave did work. So just kind of an interesting little tidbit for you. Okay, so what about for things that cannot get wet? Okay, that is for actually quite a number of things. We can use a dry heat, and there's different methods of dry heating. I think I have them on the slide here. So some things are sterilized outright by flaming. You can see in this slide here, what they're showing is an instrument that would be used to manipulate a microbial culture. So this is called an inoculation. In this case, you directly sterilize it by sticking it in the flame. And it's just a piece of metal, so it doesn't really matter. And then it gets cooled off before it touches the microbes, and that's all good. Some things are incinerated. If something is some dressings from something that is extremely dangerous, it's going to get incinerated, which means it's all burned up. Now, you can't burn everything up all the time, so it's not always the ideal instrument or the ideal method. But sometimes things are hot air sterilized. What that means is put in an oven. I told you the temperature, 120 degrees. But this method just takes a lot longer. The thing about steam really permeates things. And it permeates very quickly, and with the pressure, it can get into things very easily. Hot air, dry air is just a lot slower. I think I have a slide here that's actually showing, what did I do? Show some times. So if you take a look at this, moist heat versus dry heat, right? So Bacillus subtilis, moist heat, autoclaving, 1 minute kills it. So autoclave can be quite effective. Usually when we stick things in autoclave, the standard setting is 15, 20 minutes. It actually takes an hour because the thing has to heat up and cool down at the end. But the sterilization time is usually 15 minutes. And you can see that on this list here, all these organisms are dead within 10 minutes, right, of the autoclaving. Dry heat, on the other hand, can take much longer. You're talking about an hour to 2 hours, sometimes longer, depending on the organism, and if you're depending on what you have. So this is kind of a backup method, and sometimes it's used for certain instruments that just can't get wet. But then again, they have to be able to experience that much heat. So we can't do that with everything. There's a lot of things that are plastic as well. We can't throw in an autoclave or a dry oven. So kind of a backup plan in some cases, and for some settings, but not really that commonly used. Okay, so what about the opposite of heat? What about freezing? Not really sterilizing anything. Freezing, what you're doing is basically just turning the water into a solid. Right? You turn water into ice, and now the microorganisms, they can't do much, right? They can't metabolize because all the water is frozen. Some things will get killed. One thing that water does when it forms ice is it forms ice crystals. So this is usually enough to kill animals. There are very few animals that can survive freezing. There are a few that are specialized to survive freezing, like turtles. That's what turtles do in the winter, right? Isn't that crazy? They just burrow in the mud and they get frozen. They have special proteins in their blood that allow them to not die from that process. But most animals die, including worms. So that's why I actually have a picture of sushi. In Canada, in order to serve sushi, it has to be frozen. Now, usually we don't freeze it just by throwing it in the freezer because that affects the taste. So usually what most sushi restaurants do is it gets flash frozen. So that's using like liquid nitrogen. And because all raw fish has parasites in it. Fish, by the way, are loaded with parasites, just loaded. All sorts of worms and eggs and microscopic things that you don't know are in there. And so they get flash frozen, and this is going to kill them. It's not going to kill a lot of bacteria, not going to kill a lot of viruses, but for the case of sushi, this is usually okay. If it's kept clean and flash frozen, it's going to deal, that's going to deal with most of the microorganisms. So if you have your ground meat and you bought too much, you throw it in the freezer. That's not enough to kill the microorganisms, so don't eat your raw ground beef, please. Just not a thing. So this is just kind of preserving food and works on worms anyway. Refrigeration, same thing. You're just slowing growth down, not killing anything. Well, we keep things in the fridge. right? You get medicine, vials, all sorts of things, vaccines, they often get kept in a fridge. The idea is that it will slow down any growth if there is anything in there. And again, usually anything that's kept in a fridge has some sort of best before date, right? Kind of thing. Some things grow well in fridges, and the two things we talked about before are molds and listeria. And more of a concern for the food industry than the medical industry generally, but fridges aren't doing much. They're not sterilizing things, they're just keeping things fresh a little bit longer, just stretching out the lifespan of something. Okay, so that's heat, right? Like I said, the gold standard really is autoclaving for most things. Sometimes we can't autoclave things, or we're looking for other methods. Another common method that's used is filtration. And filtration can be used for liquids, it can be used for air, and the whole idea is you have a surface with teeny tiny holes in it. And so maybe the liquid can go through or the air can go through, but the bacteria cannot. And there's all sorts of different types of filters and filter systems out there. You can see this here on the left is a cartoon of a system that I've used in the lab. So sometimes I've had some liquid and I don't want to wait an hour for the autoclave. And it's a small amount of liquid. So I have I get a sterile flask, and you can see up here there's this special little funnel that I can put a sterile filter on. I can put a suction hose onto it, and I can bring the liquid through, and it's going to trap any bacteria in the liquid on the filter, just like is shown here in this diagram, and the liquid will pass through. There's different methods. Sometimes this is a little bit dirty, but it usually works quite well. You can see there's different sizes of filters that you can get. So most people, when they do filtration to sterilize something, they're using the 0.22 micron. So why not just use the better filter? The smaller the hole, the harder it is to push stuff through. So usually 0.22, you're getting rid of most bacteria, maybe not getting rid of viruses, but viruses aren't usually going to grow in stuff. Viruses usually need to infect a person or a cell in order to grow. Whereas bacteria, a little bit of food around, they can spoil a sample very quickly, and that will get rid of kind of most of the things you need to get rid of. Like I said, filtration is used in a lot of areas. A lot of hospitals now are starting to add filtration systems, and usually when they build a new hospital, these things are hard to add after a hospital's been built. But often, large hospitals that have special areas for certain types of patients. For example, people who are burns victims, they're very sensitive to getting infections on the skin. And so they'll have these special wards that have HEPA filters built right into the infrastructure, and it filters out the air to try to prevent any bacteria and dust particles, or at least minimize them from getting on people in these burns wards. Hard to say that, burn ward. And like I said, this is getting more common in newer hospitals. It's really hard to retrofit an old building with this kind of thing. A lot of other places, my understanding is that a lot of new swimming pools will also have filtration systems. You can imagine a swimming pool, there's moisture everywhere, right? And we want to prevent mold and whatnot from getting into things. So a lot of the air will get filtered to try to get rid of that particular issue. HEPA filters, you can see what it stands for, high efficiency particulate air. It's a special type of filter that you can get for certain applications. This one here is a biological safety cabinet. We have one of these in the biology lab, and this is a surface where we work with large cultures. So the idea behind the biological safety cabinet is that the air that is inside the cabinet gets sucked up and through a filter and doesn't get blown out and into the face of the person who's doing the work. So again, it's an extra safety precaution. You're starting to see vacuum cleaners advertised that they have HEPA filters in them. Believe it or not, I actually talked to a HEPA filter salesman, and he said that's garbage. It's not true. Those are not real HEPA filters, but I don't really know anything about vacuum cleaners, so not going to comment on that. Okay, time check here. Okay, I think we're roughly where I want to be. So what else can we do to kill these things physically? Like I said, chemicals we'll talk about next day. We can dry things out. This is a terrible method for disinfection. It's a classical method for food preservation. You can see in this case what they're showing on the picture is a bunch of fish that have been caught, and they're drying out the meat. So this works really well in some ways. You know, you're getting rid of the moisture, and the moisture is what's allowing microorganisms that are going to spoil that food to grow. But in the case of medical things, this is a really bad idea because there are many microorganisms that can survive that, particularly the endospores and certain viruses. So this is why dressings and all sorts of things, we have to autoclave them. You know, just drying things off is not going to be the best method. So just wanted to mention that one. This is the exact opposite. This is a reservoir for reinfection. Okay, so let's talk about radiation. Okay, this is something that is becoming more and more and more common because we're getting better instruments and better methods for doing so. And so it's worth it to talk about how this works. So it says here, you know, radiation is basically energy in waves. So the light in this room is radiation. Comes in different colors, but visible light is not very high energy. You know, it might be enough to warm you up on a sunny day, but it's really as you get to the smaller wavelengths that we're getting a lot more energy out of these things. So radiation can be classified as ionizing and non-ionizing. Ionizing is high energy, non-ionizing is low energy, and they both have their applications. And so it's just worth mentioning a little bit about them now. So you can see this is the spectrum. And over here on the left is low energy stuff. So things like cell phones, radios, TV signals, even microwaves are relatively low energy. Visible light is there in the middle. And on the right-hand side in the red, these are the things that actually, there's enough energy that can start damaging DNA. So this is the stuff that is useful. So the ionizing radiation, if you know what an ion is from high school chemistry, an ion is a charged particle. Ionizing radiation can actually break bonds and make ions. So that's where the name comes from, right? And so it's high energy. This includes things like X-rays, certain types of UV, right? So UV, there's a whole range of it. If you know a little bit about UV light, they talk about UVA, UVB, UVC. So B and C are the higher energy UV stuff that can cause cancer, right? And this is the stuff that we're concerned about, why you wear sunscreen and all that, and you know, if you're concerned about skin cancer. Because it can damage DNA, and damaged DNA can lead to cancer. But damaged DNA can also be good for us because we can destroy the DNA in the microbes. So high energy stuff depends on the application, but you can see there's some listed there, X-rays, cancer treatments, but you know, we can also sterilize a lot of things with ionizing our radiation as well. So this is why, you know, if you have to get an X-ray, or if you're helping someone to get an X-ray, right, you know, you're going to wear the protective vest, or you know, it has lead in it, and so that they're only X-raying the part of your body that actually needs exposure to the radiation. This is why the technician, you know, they walk around the corner out of the room because the technician is doing that all day, and he or she does not want to have exposure all the time, because excessive exposure can lead to cancers, right? So that is a concern. Something we're starting to see now is these ionizing radiations being used for sterilizing certain things. There's been a lot of this in the news in the last few years about ionizing food. And so in Canada, we're starting to use this for certain food products. And the whole idea is you have food and you want to preserve it. And unfortunately, there's fungi and other microorganisms that are going to spoil that food. So what do? You hit it with the radiation, and it's going to kill the fungi, right? And so in Canada, we actually have, I just looked this up, we have 6 types of food that are approved in Canada for food irradiation. So onions, potatoes, whole wheat flour, whole and ground spices, dehydrated seasonings, fresh and frozen ground beef. I'm not sure why those foods in particular are approved, and why not other foods. So food irradiation is actually pretty cool. It's been used in the States for quite some time now. This is the international symbol for food irradiation. In the US, I think it has to be labeled. In Canada, it's not a requirement. Some people are afraid of this because they're thinking radiation on my food is radioactive, this is going to kill me. That's not true, right? If I turn this light out, as soon as I turn the light out, there's no more light in the room, right? It's the same thing with this kind of radiation. As soon as it's turned off, the food is not irradiated anymore, right? You're not making the food radioactive. Radioactive is an entirely different type of chemistry. It's actually very safe and works really well. You can see with these strawberries here, non-irradiated and irradiated strawberries. So this is getting common, and certain types of plastics are hit with ionizing radiation as well. You can imagine all sorts of plastic things that could be used in a lab or in a hospital, syringes and petri dishes and whatnot. In some cases, they're using ionizing radiation. So the one that's used the most common in places is actually UV and UVA. Right? So UVA is actually considered to be non-ionizing. So it can still damage DNA, but in a slightly different way, and really the difference is only important to chemists, but damaging DNA. And I think I've got a slide here showing what happens, so you can see that it can make something called thymine dimers. So thymine is one of the nucleotides. Remember, there's A, T, C, and G, right? So what it can do is actually kind of swap the bonds around in the thymines, and rather than being two thymines, they kind of get stuck together. And now this is damaged DNA, and when you have damaged DNA, you have non-functional genes. So this doesn't necessarily kill the organisms, but it makes that they can't reproduce, right? So they can still metabolize, they still have some enzymes floating around their cytoplasm. That particular cell can still be alive, but the DNA is damaged to a point that it can't reproduce. And so this is really what's going on here with this kind of irradiation. And it's enough to effectively sterilize things because the organisms are, they can't reproduce, right? So like I said, UV is good for a lot of things that are plastic. Here's a small UV irradiation machine that you can buy. I think, I remember, maybe I saw this on Amazon. I was looking for it on the internet. You can buy them. There's lots of legitimate uses for these things. Like I said, plastics, petri dishes, and whatnot. You know, they come sterilized from the factories, and this is a pretty common method used for sterilizing these things. I saw. I think actually this one I saw was advertised for nail salons. So I don't know what they'd be using in nail salons. I've never been, confession, but I'm guessing scissors and clippers and things like that, right? So this is what it was being advertised for, right? I'm seeing all sorts of these things now, right? I saw this one here on the right also advertised on the internet as a personal sanitizer. And so there was a little video that went with it and showed somebody who was staying in a hotel, and they're sanitizing the remote control, you know, and some people, this is a concern, right? Some people are massive germaphobes. I'm not a germaphobe, by the way. I like microorganisms, except for like dishcloths. Like that's the one thing that I just really grosses me out. I can't touch a dirty dishcloth, by the way. Just absolutely horrifying. You can see this germicidal lamp as well. So McDonald Island has these, again, part of the ventilation to kill any spores and fungi that are going to cause mold. Also used in a lot of hospitals now. Newer buildings are starting to retrofit these things. Okay, so that are the physical methods. So kind of the walkaway method, or the walkaway message here, is that autoclaving is awesome. When in doubt, you're autoclaving something. If you can't, UV is pretty good, and they're pretty cheap. UV sterilizers are not that expensive, and so that would include things that are can't be exposed to heat. A lot of plastic, for example, will melt. A lot of things can't get wet, so you might use UV as well. Okay. The other methods kind of have their uses. Like I said, filtration has its usage for structural buildings. Filtering out the air to minimize infections and whatnot, and getting more common. But you're not going to retrofit a whole building that's going to be millions of dollars. So it's not in full use yet, except for newer buildings. And all these other things, like I said, lots of uses in the food industry. This doesn't really cover everything in every scenario, and this is why next day we're going to talk about chemicals and disinfection and how that's going to kind of complement our physical methods here. So that's it for today. Don't forget to show up on Thursday at 10:30 instead of 10. And I guess have a great week.